Welcome to this explainer on general microbiology. Understanding the unseen microbial world isn't just an academic exercise. It's the absolute foundation of infectious disease diagnostics and treatment in veterinary medicine.
The topics covered today include an introduction to microbiology, historical development and theory, biological classification, microbial morphology, and finally, microscopy and scale.
Starting with Section 1, Introduction to Microbiology and Foundational Concepts.
Microbiology is strictly defined as the scientific study of microscopic organisms. This means analyzing biological agents that are simply too small to be observed clearly by the unaided eye. There are, however, some rare and fascinating macroscopic exceptions, like the massive bacteria Theomargarita and Epilopetium, which completely break this conventional rule.
Microorganisms exist in a delicate balance regarding their impact on higher life forms. On one side, microbes maintain absolutely indispensable ecological roles. They actually contain 50% of biological carbon and 90% of biological nitrogen on Earth. They are essential for nutrient cycling, digestion in animals, and the industrial production of things like vaccines and antibiotics. Conversely, a specific subset functions as pathogenic organisms, capable of inducing clinical disease by damaging tissues in animals and humans. Recognizing this dual nature and accurately identifying the pathogens is paramount for veterinary science. Moving on to Section 2, Historical Development and Theory, charting the path from philosophy to experimental proof. For centuries, biological origins were fundamentally misunderstood. The theory of spontaneous generation, initially proposed by Aristotle, suggested that living organisms could develop directly from non-living matter. This theory was systematically dismantled over time. Francesco Redi and Lazzaro Spallanzani conducted carefully controlled experiments involving decaying meat and heat-sterilized mediums in sealed flasks, showing that microbial growth required external exposure. Later, Theodor Schwann, alongside scientists like Schroeder and von Deutsch, filtered air through red-hot tubes or sterile cotton wool. This proved that sterile mediums remained perfectly clear even when exposed to treated air. The final definitive proof against spontaneous generation was delivered by Louis Pasteur. Often recognized as the father of microbiology, he made this monumental contribution using a uniquely designed swan neck flask. By strictly controlling the variable of airborne dust, this step-by-step methodology definitively proved that microorganisms arrive from external contamination, not spontaneous generation. Pasteur boiled a nutrient solution, leaving the flask open to the atmosphere. Crucially, that curved neck trapped the dust and germs, and the broth remained entirely sterile. However, the moment the neck was broken, exposing the broth directly to falling debris, microbial growth commenced immediately. A brilliantly simple yet revolutionary experiment. With spontaneous generation disproved, the germ theory of disease took root. To prove exactly how microorganisms cause clinical disease, Robert Koch formalized a sequential methodology using the anthrax-causing bacteria, Bacillus anthracis. Known as Koch's postulates, this framework demands 4 strict steps. First, the specific microorganism must be present in every single case of the disease. Second, it must be isolated and grown in pure culture. Third, inoculating this culture into a healthy host must replicate the original disease. And finally, the identical microorganism must be re-isolated from the newly diseased host. Next is Section 3, Biological Classification and Taxonomy. Cellular life diverges structurally into 2 main categories, eukaryotes and prokaryotes. Eukaryotes, which include animal and fungal cells, are typically much larger. They possess true nuclei bounded by membranes, multiple linear chromosomes, and complex organelles like mitochondria and Golgi apparatuses, and they replicate via mitosis. In sharp contrast, prokaryotes, which include all bacteria, lack true membrane-bound nuclei. Their genetic information is typically held in a single circular chromosome, and they replicate through a much simpler process called binary fission. Modern classification utilizes phylogenetic analysis of ribosomal RNA, revealing 3 primary domains of life: archaea, bacteria, and eukarya. An interesting concept here is the endosymbiosis hypothesis. It suggests an evolutionary bridge, proposing that eukaryotic cells originally acquired organelles, like mitochondria and chloroplasts, by engulfing primitive prokaryotic cells. Orderly classification relies on a strict hierarchical system. The levels ascend from species up to genus, family, order, class, phylum, and finally kingdom or domain. This culminates in binomial nomenclature,
providing standardized Latinized names for precise scientific communication. For example, Bacillus anthracis identifies both the genus, Bacillus, and the specific species, Anthracis.
Section 4 covers microbial morphology and the incredible diversity of these agents. Distinct morphological features differentiate the major classes of infectious agents. Bacteria typically possess rigid cell walls containing peptidoglycan and appear in forms like spherical cocci, rod-shaped bacilli, or helical spirochetes. Fungi, on the other hand, present as either single-celled yeasts reproducing by budding or multicellular molds forming branched structures called hyphae. Stepping entirely away from cellular life, viruses are completely acellular. They consist merely of DNA or RNA enclosed in a protein capsid and absolutely require a host cell to replicate. Finally, prions challenge the standard biological definition altogether. They are completely devoid of nucleic acid, consisting solely of abnormally folded proteins that are highly capable of devastating neural tissue. And finally, Section 5, Microscopy and Scale. Specific laboratory techniques are deployed based on the exact properties of the target microorganism. Bright field microscopy is the standard for stained bacteria. Dark field microscopy scatters light to illuminate fine, unstained microorganisms, like spirochetes against a completely dark background. Fluorescence microscopy utilizes specific antibodies conjugated with fluorochromes to perfectly identify particular pathogens. For subcellular details and viruses, electron microscopy is strictly required, using electron beams rather than light to achieve staggering magnifications exceeding 100,000 times. To properly grasp the microscopic scale, a universal baseline is necessary. Consider a typical mammalian red blood cell, which measures approximately 7 micrometers across. This eukaryotic cell is readily observable using conventional light microscopy. Shrinking the scale slightly, a typical rod-shaped bacillus bacterium measures 5 micrometers. While still visible under light microscopy, observing most bacterial cells clearly requires an oil immersion bright field objective, operating at a magnification of 1,000 times. Descending deeper into the submicroscopic realm, the scale shifts from micrometers to nanometers. The poxvirus, one of the larger viral structures, measures a mere 300 nanometers. At this tiny size, it is completely invisible to conventional light microscopy, necessitating the use of electron microscopy. At the extreme lower limit of the viral scale lies the parvovirus. Structurally reduced to just 20 nanometers across, it represents one of the smallest entities capable of inducing profound clinical disease in veterinary medicine. This microscopic descent naturally leads to a profound frontier in general microbiology. The existence of nearly indestructible prions challenges traditional biological paradigms. Since they are physically smaller than the 20-nanometer parvovirus and lack DNA entirely, detecting and neutralizing these abnormally folded proteins before they cause irreversible transmissible spongiform encephalopathies remains a massive open question. If traditional methods target nucleic acids and cellular structures, how exactly must diagnostic technology evolve to combat non-living, non-viral pathogens? That question will dictate the next great chapter in veterinary medicine.